I hope we will have some time at the end for a plenary discussion, but we'll see. We'll now move on with Fabian Kiessling. He's the head of the Institute of Experimental Molecular Imaging in Aachen. We discuss a lot about the principles of targeting, right? Also why nature targets and how nature targets. I'm not really sure what he's going to present now, but I'm very much looking forward to your talk. So,
yeah, thank you very much for the invitation and for the introduction, Twan. I think we already had a terrific start of the session with a great talk where I can only agree on all the points that were made. Since I'm a physician, I come from another angle. And in the next minutes, I would like to share with you some important points that I usually take into consideration before I start a project on a new diagnostic or therapeutic drug. And in this context, I think we have to think more critically on some preconditions that we want to have from diagnostics or therapeutics. So I may start with the diagnostics. The most important thing is that they will provide us with really relevant information that's clear. But what does relevant mean? Relevant with respect to diagnostics does not mean to have a nicer image or to see a certain receptor. No, we want to change the therapeutic conduct of the patient. We want to improve the therapeutic outcome of the patient with a diagnostic drug. If this is not given, and if you do not have a chance to fulfill that, you can forget about the development of a new diagnostic tool. Then the next thing is that the imaging modality that we choose has to be suitable for the clinical purpose. So, for example, I see many papers where people use optical imaging or ultrasound in mice for whole body staining. This may work in the mouse, but if you consider the penetration that you need in a human, this may not work in a clinical scenario. That means that this kind of development is not translatable. Then clinicians usually want to have target accumulation within minutes because they want to longitudinally scan the patients, and everything that takes long is difficult to integrate into clinical routine because the patients then have to come twice or they have to stay long in the hospital. The next point is that we really need sensitive and specific detection. If we talk about targeted diagnostics, for example, we have to consider that we don't want to see the drug or the diagnostic at the end. We want to get information about the degree of receptor expression. And this also demands on having a very rapid exchange between compartments, on a very rapid elimination of the unbound fraction from the blood and from the body. And of course, if we talk about diagnostics, it must be ensured that there are no side effects. I don't talk about low side effects, I talk about no side effects, because we are also treating healthy subjects, and they have to have a life expectancy which is quite normal over several decades of years. It's a little bit different if we talk about therapeutics. There, of course, the most important point is high therapeutic efficacy and the decrease of side effects, as we have it from many nanomedicines, which we can achieve by decreasing the uptake in healthy organs. Usually, in contrary to diagnostic agents, where we usually want to have a short blood half-life, here we want to have a long blood half-life to really use the EPR effect. And, of course, we also need strong and long target accumulation. We don't care in this case whether it's a receptor-specific information. The only thing that is really important is that the drug at the end gets into the cell that should be targeted. And if we consider these points, we have to consider the renal elimination threshold, which is about at 7 nanometers, the size of albumin. And we have to take into consideration that everything that is smaller will be eliminated by the kidney, thus may have a short circulation time, but everything that is larger, except is rapidly taken out by the reticular endothelial system, will circulate for longer periods of time and may thus be better useful as an EPR-based drug delivery system. But still, there's a big range in sizes that we have to consider, starting from 10 nanometers up to 1,000 nanometers. And the question is really which size is ideal if we want to develop a drug delivery system, and which size is ideal to do active targeting. In this context, I come back to the point that was made by our previous speaker. We should also consider the size of antibodies, and I think it has a clear reason why nature made the immunoglobulin G, for example, with a size of 10 to 50 nanometers, and the first phase antibodies with a size of 30 to 35 nanometers. Well, there were some nano-based diagnostics implemented in clinics, and with this slide I also want to show you how you have to think also about alternative strategies. Since many of the nanoparticles are internalized by the reticular endothelial system, the first diagnostic agents were used to negatively contrast these tissues and thus to get better contrast to malignancies either in the liver or, for example, in lymph nodes. But this agent was withdrawn from the market several years ago and replaced by this small gadolinium agent called gadolinium EOB DTPA, which is known under the trade name Primovist. And the reason is quite simple. First of all, the elimination from the liver is much faster of this gadolinium chelate, what is taken up, by the way, by the hepatocytes. That makes repetitive imaging much easier, which is, of course, appreciated by the medical doctors. The second point is that the medical doctors prefer a positive over negative contrast. And the third point is that this product is much cheaper to produce. A similar reason led to the withdrawal of these dendrimeric MR contrast agents, which were designed for angiography purposes. You see here below an image of gadomer 17, which is this dendrimer 17 gadolinium chelates at the end, which provided really excellent angiography images, not only in animals but also in early human trials. But also this one was finally replaced by a small gadolinium agent, which has a strong binding affinity to albumin and thus makes the same job by enhancing the blood pool. Well, still we may use nanoparticles for imaging EPR, but it's still an open question whether we really need the information about EPR for personalization. Certainly it's very heterogeneous between patients. I agree, and I also believe that there is a need for biomarkers, but you should also consider that we don't want to predict the accumulation of the drug. At the end, we want to predict the response of the patient, and for that purpose, we have to ensure that the accumulation equals to the response. We will come later back to that point. The next question is, what is the right imaging strategy to predict EPR? Do we really need a nanoparticle for that, or a theranostic agent, or can we get the same information already with clinical procedures that are already in use, like a scan with, for example, a small gadolinium chelate and a dynamic imaging procedure? Well, in the next point, I would like to discuss a little bit about active targeting. And active targeting has something to do also with barriers, because between your probe and the target are usually biological barriers that you have to cross. And there are many barriers depending on the target. The first one is the blood itself. The second one would be, for example, the endothelial layer. Then you have to cross the stroma with the cells that are within the stroma. Then you may have to cross a cell membrane. You have to cross a cytoplasm. You may have to cross a nuclear membrane, depending on the target. But what you have to keep in mind is that the more biological barriers exist, the less probe will reach the target, the more unspecific background will occur in each of the compartments. And if you want to make a diagnostic probe that gives you information about a certain receptor, then the probe must be very, very small or smaller with the increase of the number of barriers that are between the injection site and your target. For targeted therapeutics, I don't care so much about the background. Of course, I care about the compartments, but only important thing is that after a certain time, the target is reached, so I can live with a certain degree of background. But here I have to balance very carefully between the EPR-based accumulation, the tissue penetration, and the active targeting capabilities. Thus, it's not surprising that so far there was only one publication of a targeted diagnostic nanoparticle in human use, and this was published by Phillips et al. 2014 in Science Translational Medicine. And based on the things that I said before, you will not be surprised that the particle that they were using was very small, only 2 to 3 nanometer large. It was a carbon dot, dual functional for PET imaging and subsequent fluorescent guided surgery, targeting alpha V beta 3 integrins, and this also showed a very rapid renal elimination. What about targeting larger particles? And there was already in 2006 a nice paper by Kirpotin who targeted liposomes, and you see here the accumulation curve, and you see that the difference between the targeted and non-targeted probes is very little. Certainly you will agree with me that this is not suitable for a targeted diagnostic approach, but maybe it is still valid for therapeutic purposes, but certainly not for the part of accumulation. Knowing these facts, we thought maybe we then should target not targets that are behind several barriers, but we should target something that is already occurring after one barrier. And so we thought, okay, let's take a polymer that is targeted against angiogenesis and that accumulates very nicely via EPR, then we should have an added effect. Yeah? And indeed, so we took the polymer, we functionalized it with NGR so that it binds to aminopeptidases, or with RGD so that it binds to integrins, and compared the accumulation with the control polymer. And indeed, you see that the active targeting brought us an early, stronger accumulation. But then on the long run, you see that the area under the curve of the targeted probe is smaller than of the non-targeted one. And the reason is very simple. If you have a system that is optimized for stealth properties, it will strongly accumulate via EPR. And the blood half-life is the main determinant for EPR-based accumulation. If you now bind the targeting unit to it, you opsonize it, for example, to macrophages and other cells, they will take it out of the blood, so the blood half-life will decrease, so the EPR-based effect will also decrease. And at the end, although you have a successful targeting at the beginning, you may end up at the end with a lower accumulation of the probe than with an untargeted control. Based on that, we thought, okay, we want to do active targeting, and we want to target outside the vascular system. Maybe for these kind of drug delivery systems, we have to go to smaller sizes. Maybe we have to learn from nature about these antibodies, which are really optimized for long circulating properties, for extravasating in tissues with altered endothelial leakiness, and which should also well penetrate the tissue. And as a target for that approach, we have chosen the riboflavin transporters RFT1, 2, and 3, which are known to be overexpressed not only by tumor cells, but also by other cell compartments of the tumors, like the endothelial cells, the macrophages, and there's even an interstitial soluble protein that keeps the riboflavin trapped inside the tumors. As drugs, we designed 2 model drugs consisting of star packs here, with 2 riboflavins on 3 arms and a fluorescent dye for detection. We designed them with a size of about 5 nanometers and 50 nanometers. This would be the size usually of an IgG. Of course, you have to take into consideration the different sterical properties, but well, approximately it should fit. Yeah. If you now compare the blood half-life of these probes, you see that with a small one, we have a blood half-life of 1 hour because it's linearly cleared. With the other one, we have a blood half-life of 13 hours. And how does that affect the binding that we can observe? And that's very interesting. We evaluated these probes in 2 tumor models, one that is very angiogenic, the A431 tumors, one that is very rich of stroma, the PC3 prostate cancer model. And as you can see, the EPR effect led to a very strong accumulation of the bigger probe, much more than of the small targeted one. You also see that the active targeting did only improve the accumulation, in principle the retention of the small probe, but had not a significant impact on the accumulation and retention of the large probe. But if you now look to polymer internalization into the cells, situation is different. Here you see that although the overall accumulation was low for the small one, we have higher internalization, even higher than of the targeted large one, at least in one of the tumor models. But you still see that the active targeting has clearly improved the cellular uptake, not only of the small one, but also of the large one, so that you can say you can at least bring more of the probes into the tumors. We could also show that already this small size change from about 5 to 50 nanometers changed also the preference of the particles to certain cell types. So while the small ones were mostly taken up by tumor cells, followed by macrophages, followed by endothelial cells, the larger ones were mostly taken up by macrophages because they lay longer in the interstitial space, and then by tumor cells, and almost nothing ended up in endothelial cells. So I may summarize my findings. So I think it's very important to carefully consider pharmacokinetic properties and biological barriers in the design of diagnostic and therapeutic agents. I think it's very important to consider that not every imaging modality is really useful for every medical question and not sensitive enough to image every target. I think if you really aim for extravascular molecular imaging, your probes should be very small, clearly smaller than 5 nanometers. However, I see clear indications also for larger nanoparticles, for example, for vascular imaging, for MPS imaging, for cell and tissue labeling, and for EPR assessment. Concerning therapeutics, I think we can distinguish 2 kinds of drug delivery systems. The first, that is accumulating via EPR and then releases the drug in the interstitial space, or that are taken up by macrophages that then die and release the drugs that then diffuse to the cancer cells. I think these really need some degree of personalization. However, the right imaging biomarkers still need to be found. And then we have drug delivery systems that really need to be internalized by a certain target cell. And these may be drug delivery systems that carry highly toxic drugs, that may carry drugs that not diffuse to target cells, or drugs that are not internalized by the cells, or drugs that should be directed to a cellular subcompartment. And here we really may need active targeting, and my hypothesis is that these drug delivery systems should be smaller than the majority of nanosystems that are available by now, and that we really need an optimized balance between active and passive targeting and tissue penetration. In this context, with respect to active targeting, we should keep in mind thus active targeting will not improve the accumulation of nanomedicines, but only retention and internalization, that it will be less successful with DDS that show low tissue penetration, and that tumor accumulation is not automatically equal to internalization and therapeutic efficacy. With these words, I end my talk. I see that my time is up, but I hope that we still have time for a few questions. Thank you very much.
Thank you, Fabian. Let me start by sharing that we have good and bad news. The good news is that Gooitsen van Dam did make it. He had problems with his train, but he is now present. He will give his talk. And the bad news is that that means that everybody really needs to stick to the time. We only have brief time for discussion. So I would say we have time for 1 or 2 questions at this stage. Yes?
Maybe just a comment, because we talked about that also last year. Developing theranostic agents where you can actually quantify also the amount of nanoparticles that get into the tumor, they stay there, they are retained there. And maybe this curiosity can also involve the recent experience by Hazy on zirconium-labeled liposomes. What is the increasing cost in developing a liposome per se or a nanoparticle per se as a drug delivery agents versus having a real theranostic agent?
I don't know. I would even go one step farther and say, what is the advantage of these kind of agents in comparison to standard clinical procedures like DCE MRI or just a perfusion scan with CT or ultrasound? This is an open question. It has not been systematically evaluated yet. Nobody really made the comparisons whether, for example, the accumulation of a small agent, if it is considered in an appropriate pharmacokinetic analysis, will give us the same information that we may get from a zirconium-labeled liposome, for instance. I think these are really important studies that have to be made in the future and where we need more data.
I think that we need to stratify the patients for that specific nanomedicine.
I don't know whether this is really necessary.
And I think some of the data, as well as some of the conclusions that you presented, are actually aiming in that direction. It might still be a matter of discussion, but I think that we really need to look very carefully into that. And if the difference in cost between having just a drug delivery system versus a theranostic system is not that dramatic, I think we should really look into that very carefully. I don't know if Hazy can actually comment on that. I've seen his hands on.
Well, we have to understand more about the biology. You see, if we, for example, would know that the collagen content in a tumor is a major factor that drives the success of a nanotherapy, it might be enough to have a companion diagnostic in a general means that quantifies collagen content. Then I would not need any theranostic agent for that purpose, and it would be much more cost efficient for a company to develop such kind of diagnostic tool. We just don't know it yet. To which level of detail we have to come down really to predict patient response.
So I suggest we pick that up after Daryl's talk, because we'll talk about the companion diagnostic and the theranostic approach, I guess. So that's the unspecific versus a specific version. Hezi, very last short comment.
I think especially first start with you, the added cost should be minimal. If you have the system to run the zirconium, you have all the facility, the added cost is nothing, because you mix the two in the presence of something, and in a matter of 1 hour incubation, that we know under this condition no drug is leaking, everything was tested, you have the things ready to inject. So you have nothing to do, basically. Now, I agree with you. The bottom line is the efficacy on the patient. The efficacy required to understand many things. One of them is how much of your nanosystem is reaching the tumor. But that by itself is not enough. It actually may be misleading, not only not enough. So I think we need to study the efficacy. And I think still you can do it if you do a systematic theranostic analysis, namely the first injection, then you follow up. You can see how the tumor really changed, and you can learn about it. But of course, if you know about the tumor microenvironment in terms of how the liposome move inside, we did some work recently, published a year ago, I think, about some large polymer and pancreatic tumor that actually measured diffusion of the small, low molecular weight drugs inside. That's really the most important. Is if you don't build your system on the fact that the liposome will be taken up by the cell, but on the drug release, you have to know the distribution of the drug in the tumor and how much of it is taken up and how much resistance you have. So there are so many questions to answer. But the first thing, to know how much is reaching the tumor. If nothing reaches the tumor, then you don't get efficacy. Yeah. This was straightforward. If a lot get, you still don't know if you get efficacy.
Yeah. Figure out how we can decide whether something is going to the tumor or not. And if nothing is going to the tumor, those patients might not respond and they should be excluded. But then there are many more questions to answer.
That's right.
Yeah.
Maybe I fully agree. I fully agree with you, but only one remark about the zirconium. Indeed, it's not more expensive to develop such kind of theranostic approach, but you also have to think about the treatment costs in the clinic. If I can do an ultrasound scan with contrast agent, which costs me 100 euros, yeah, or a dynamic contrast enhanced MRI scan that costs me 200 euros, in comparison to an 800 euro PET scan, the insurance agencies will certainly, if the two methods are equivalent, approve the one that is cheaper. And it's also not the production costs that are connected to the theranostic. It's also the daily costs that are coming up in the clinical routine. And these may be, for such kind of PET approaches, quite high.
Yeah. You know, if you're wasting your drug that costs, I don't know, a few $1,000s per injection against the money that the test costs, it becomes ridiculous. So you have to do the best test in order to see if your drug has a value, and that's the major save.
Absolutely, absolutely. You're absolutely right for that.
Good. With that, we move on. Thank you, Fabian.